The germ theory of disease stands as a monumental pivot in human history, reshaping our understanding of life and death. It revealed that the smallest of creatures can wield the greatest of powers over human fate. And so, we embark on a journey to uncover the hidden world of microbes, where the very fabric of our existence hangs in the balance. In the sweltering summer of 1837, a young French chemist named Louis Pasteur stood at the threshold of his academic career, unaware that his fascination with the mysteries of fermentation would soon propel him into a lifelong quest to unravel the enigma of disease. The air was alive with the hum of insects and the sweet scent of blooming flowers in the gardens of the École Normale Supérieure in Paris, where Pasteur had just begun his studies. It was an era marked by great scientific upheaval, as the old certainties of spontaneous generation and the concept of a static, unchanging natural world were being challenged by a new wave of thinkers. As Pasteur delved into the world of chemistry, he found himself drawn to the work of Antoine Lavoisier, the father of modern chemistry, who had once walked the same streets of Paris. Lavoisier's groundbreaking discoveries on the nature of elements and compounds had laid the foundation for a new understanding of the natural world, and Pasteur was determined to follow in his footsteps. The young chemist spent countless hours poring over the works of Lavoisier, as well as those of other prominent scientists of the time, including Georges Cuvier and André-Marie Ampère. Meanwhile, across the English Channel, a British surgeon named Joseph Lister was making waves in the medical community with his pioneering work on antiseptic surgery. In the bustling hospitals of London, Lister was confronted with the grim reality of post-operative infections, which often proved fatal to his patients. His quest for a solution would lead him down a path of discovery that would ultimately change the face of medicine forever. As the Industrial Revolution gained momentum, urban centers like Paris and London were experiencing rapid growth, with people flocking from rural areas to seek work in the factories and mills. This mass migration led to overcrowding, poor sanitation, and a surge in diseases like cholera, typhoid, and tuberculosis. The streets of Paris, once famous for their beauty and charm, were now filled with the stench of garbage and human waste, creating an environment ripe for the spread of disease. It was against this backdrop of scientific curiosity and societal upheaval that the germ theory of disease began to take shape. In the early 19th century, the prevailing view of disease was rooted in the concept of miasma, which held that illnesses were caused by a noxious form of bad air that arose from decaying matter. This idea had been around since ancient times and was still widely accepted by the medical community. However, a growing number of scientists and physicians were beginning to question this theory, seeking alternative explanations for the causes of disease. One such pioneer was Agostino Bassi, an Italian scientist who had discovered that a fungus was responsible for a disease affecting silkworms. His findings, published in 1835, would soon inspire others to explore the role of microorganisms in causing disease. As Agostino Bassi's discovery of the fungal disease affecting silkworms began to gain attention, it sparked a wave of interest among scientists to explore the role of microorganisms in causing disease. One such scientist was Justus von Liebig, a German chemist who had been studying the process of fermentation. In 1839, Liebig proposed that fermentation was a chemical process rather than a biological one, and that it was caused by the action of chemical agents rather than living organisms. However, this theory was soon challenged by Pasteur, who had been conducting his own experiments on fermentation. Pasteur's work on fermentation led him to conclude that it was indeed a biological process caused by the action of microorganisms such as yeast and bacteria. He demonstrated that these microorganisms were responsible for the conversion of sugars into alcohol and carbon dioxide, and that they could be killed by heat or other means, thereby preventing fermentation from occurring. This discovery had significant implications for the understanding of disease, as it suggested that microorganisms could play a role in causing illness. Meanwhile, in England, Joseph Lister was continuing his work on antiseptic surgery, inspired by the discoveries of Pasteur and others. In 1865, Lister read about Pasteur's work on germ theory and realized that it had significant implications for his own work. He began to experiment with the use of carbolic acid as an antiseptic, using it to clean wounds and surgical instruments. The results were dramatic. with a significant reduction in post-operative infections and deaths. Lister's work on antiseptic surgery was soon recognized by the medical community, and he became a leading figure in the development of modern surgery. His use of carbolic acid as an antiseptic paved the way for the widespread adoption of germ theory in medicine, and his work had a profound impact on the treatment of disease. As news of Lister's successes spread, it sparked a wave of interest among scientists and physicians to explore the role of microorganisms in causing disease. One such scientist was Robert Koch, a German physician who had been studying the causes of anthrax. In 1876, Koch demonstrated that anthrax was caused by a specific type of bacteria, which he isolated from infected animals. He then used this discovery to develop a series of criteria known as Koch's postulates for determining whether a microorganism was responsible for causing a particular disease. These postulates, which included the requirement that the microorganism be present in all cases of the disease and that it be possible to isolate and culture the microorganism, became a cornerstone of modern microbiology. As Robert Koch's postulates gained widespread acceptance, they became a crucial tool for scientists seeking to understand the causes of various diseases. One such scientist was Louis Pasteur, who had been working tirelessly to develop vaccines against rabies and anthrax. In 1881, Pasteur conducted a series of experiments on anthrax, using Koch's postulates to demonstrate that the disease was indeed caused by a specific type of bacteria. He then developed a vaccine against anthrax, which he tested on sheep and other animals with remarkable success. Meanwhile, in England, Joseph Lister continued to refine his techniques for antiseptic surgery, using carbolic acid to clean wounds and surgical instruments. His work had a profound impact on the treatment of disease, and his use of antiseptics became widely adopted by surgeons across Europe and beyond. In 1879, Lister was appointed as the professor of surgery at King's College Hospital in London, where he established a reputation as one of the leading surgeons of his time. As the germ theory of disease gained wider acceptance, it sparked a wave of interest among scientists to explore the role of microorganisms in causing illness. One such scientist was Emil von Behring, a German physiologist who had been studying the causes of diphtheria. In 1890, von Behring discovered that diphtheria was caused by a specific type of bacteria, which produced a toxin that was responsible for the symptoms of the disease. He then developed an antitoxin against diphtheria, which he tested on animals with remarkable success. Von Behring's discovery sparked a new era in the development of vaccines and antitoxins, as scientists began to explore the role of microorganisms in causing a wide range of diseases. In 1892, the Russian scientist Elie Meshnikov discovered that white blood cells played a crucial role in the immune system, engulfing and destroying foreign particles and microorganisms that entered the body. This discovery helped to shed new light on the mechanisms of immunity, and it paved the way for further research into the causes of disease. As the 19th century drew to a close, the germ theory of disease had become widely accepted by the scientific community. The work of scientists such as Pasteur, Koch, Lister, and von Behring had helped to establish that microorganisms played a crucial role in causing many diseases, and it had paved the way for the development of vaccines, antitoxins, and other treatments. In the late 19th century, a new generation of scientists began to explore the role of microorganisms in causing disease. One such scientist was Paul Ehrlich, a German chemist who had been studying the properties of dyes and other chemicals. In 1909, Ehrlich discovered that certain dyes could be used to selectively target and kill specific types of bacteria, and he developed a series of compounds that were effective against syphilis and other diseases. Ehrlich's discovery sparked a wave of interest among scientists to explore the properties of chemicals and their potential uses in medicine. In 1910, the American scientist Peyton Roos discovered that certain types of cancer were caused by viruses, which were previously unknown to be associated with the disease. This discovery helped to shed new light on the causes of cancer, and it paved the way for further research into the role of microorganisms in causing illness. As the 20th century progressed, the discovery of viruses as a cause of cancer sparked a new wave of research into the role of microorganisms in causing disease. In 1913, the American scientist Frederick Tort discovered that viruses could be filtered out of bacterial cultures, providing further evidence for the existence of these tiny infectious agents. This breakthrough led to a greater understanding of the mechanisms by which viruses cause disease and paved the way for the development of vaccines against viral infections. Meanwhile, in the field of bacteriology, scientists such as Felix de Herelle were making significant strides in the discovery of bacteriophages, viruses that specifically target bacteria. In 1915, Darrell discovered that these bacteriophages could be used to treat bacterial infections, and he developed a number of phage-based therapies that showed promising results. Although the use of phage therapy eventually fell out of favor in the Western world with the advent of antibiotics, it continued to be used in some parts of the world, such as the Soviet Union, where it remained a staple of medical treatment for many years. The discovery of penicillin by Alexander Fleming in 1928 marked a major turning point in the history of the germ theory of disease. Fleming's observation that a mold had contaminated one of his bacterial cultures and that the mold had killed off the surrounding bacteria led to the development of the first antibiotic. Initially, penicillin was used to treat a range of bacterial infections, including pneumonia and septicemia, with remarkable success. However, as the use of penicillin became more widespread, scientists began to realize that bacteria were developing resistance to the antibiotic, a phenomenon that would become a major challenge in the years to come. In the 1930s, scientists such as Gerhard Domag and Selman Waxman made significant contributions to the development of new antibiotics. Domag's discovery of the antibacterial properties of Prontosil, a synthetic dye, led to the development of a range of sulfa-based antibiotics that were used to treat a variety of bacterial infections. Waxman's work on the isolation and characterization of streptomycin, an antibiotic produced by the bacterium Streptomyces griseus, led to the development of new treatments for diseases such as tuberculosis. As the Second World War drew to a close, the germ theory of disease had become firmly established as a cornerstone of modern medicine. The discovery of antibiotics and the development of vaccines against infectious diseases had saved countless lives and revolutionized the practice of medicine. However, despite these advances, scientists were aware that there was still much to be learned about the causes of disease, and that new challenges, such as the rise of antibiotic resistance, would need to be addressed in the years to come. In the post-war period, scientists such as Jonas Salk and Albert Sabin made significant contributions to the development of vaccines against viral diseases. Salk's development of an inactivated poliovirus vaccine in the 1950s led to a major reduction in the incidence of polio, while Sabin's development of a live attenuated vaccine provided long-term immunity against the disease. The success of these vaccines sparked a new wave of research into the development of vaccines against other viral diseases, including measles, mumps, and rubella. The discovery of the structure of DNA by James Watson and Francis Crick in 1953 marked a major turning point in our understanding of the mechanisms of life. The realization that genetic information was encoded in a sequence of nucleotides in DNA led to a greater understanding of the ways in which microorganisms cause disease and paved the way for the development of new treatments and therapies. As scientists began to explore the molecular basis of infectious disease, they were able to develop new diagnostic tools and treatments that targeted specific molecular mechanisms rather than simply relying on broad-spectrum antibiotics or vaccines. The 1960s saw a major expansion in our understanding of the role of microorganisms in causing disease, with the discovery of new pathogens such as the Epstein-Barr virus, which was identified as the cause of infectious mononucleosis. The development of new diagnostic techniques, such as electron microscopy and molecular biology, allowed scientists to detect and characterize microorganisms with greater precision than ever before, leading to a greater understanding of the causes of disease and the development of new treatments. As the 1970s unfolded, the germ theory of disease continued to evolve with advances in molecular biology and genetics. The discovery of restriction enzymes by Werner Arber, Daniel Nathans, and Hamilton Smith in 1970 revolutionized the field of genetic engineering, enabling scientists to manipulate DNA sequences with unprecedented precision. This breakthrough led to the development of recombinant DNA technology, which allowed for the creation of novel vaccines and therapeutic agents. One notable example of this technology's potential was the development of the hepatitis B vaccine by Baruch Blumberg and Irving Millman in the late 1970s. By using recombinant DNA techniques to produce a safe and effective vaccine, they significantly reduced the incidence of hepatitis B worldwide. This achievement marked a major milestone in the fight against infectious diseases and demonstrated the power of molecular biology in preventing and treating illnesses. The 1980s saw the emergence of new challenges, including the rise of HIV/AIDS, the identification of the human immunodeficiency virus by Luc Montagnier and Robert Gallo in 1983 marked a crucial turning point in the understanding of this disease. As researchers like David Ho and John Mellors worked to develop effective treatments, they encountered significant obstacles, including the virus's remarkable ability to mutate and evade the immune system. Despite these challenges, scientists made steady progress in developing antiretroviral therapies, ART, that could slow the progression of HIV infection. The introduction of AZT, zidovudine, in 1987 marked the first major breakthrough in HIV treatment, followed by the development of combination therapies like HAART, highly active antiretroviral therapy, in the mid-1990s. These advances transformed the management of HIV/AIDS, turning a once fatal diagnosis into a manageable chronic condition. The 1990s also witnessed significant advances in our understanding of the human microbiome and its role in health and disease. Researchers like Jeffrey Gordon and Stanley Falco began to explore the complex relationships between microorganisms and their hosts, revealing the intricate networks of interactions that shape our immune systems and influence our susceptibility to illness. As the 21st century dawned, the germ theory of disease continued to evolve in response to emerging threats like SARS, avian influenza, and Ebola. The development of new diagnostic tools, such as PCR, polymerase chain reaction, and next-generation sequencing, enabled scientists to detect and track infectious agents with unprecedented speed and accuracy. The COVID-19 pandemic, which began in late 2019, marked a major test of the global health community's ability to respond to emerging infectious diseases. The rapid development and deployment of vaccines, like those produced by Pfizer, BioNTech, and Moderna, demonstrated the power of modern biotechnology in preventing and controlling infectious diseases. As scientists like Catalin Carrico and Drew Weissman worked to improve vaccine design and delivery, they built upon a century of advances in the germ theory of disease, from the pioneering work of Louis Pasteur to the cutting-edge technologies of the present day. The ongoing struggle against antimicrobial resistance, fueled by the overuse and misuse of antibiotics, highlighted the need for continued innovation and stewardship in the face of evolving microbial threats. Researchers like Jennifer Doudna and Emmanuelle Charpentier, who developed the CRISPR-Cas9 gene editing tool, opened up new avenues for exploring the genetics of microorganisms and developing novel therapeutic strategies. Thank you for joining me throughout this historical journey. That's all for today's History with Herodotus. Remember to like, share, and subscribe. And until next time, remember that, to understand the present, we must first understand the past.